about the physical methods of gene transfer. The first important physical method of gene transfer is electrophoresion. Okay, electrophoresion, it's a simple technique, simple and rapid technique for introducing the genes into the cells from various organisms. Here what happens is, so it's very simple, okay, and here what we are going to do, we are introducing the genes into the cells, that is host cells. It may be microorganism, plant cells and animal cells. Okay, so what we are going to do is, the principle behind this technique is, in this technique we are going to make the pores, or pores are created in the cell membrane. In this technique, the pores means holes are created in the cell membrane through subjecting cells into high voltage. Okay, when you subject the cells into high voltage, what happens in the cell membrane? You will be having the membrane. So membranes will open up. Okay, when the membranes will open up, pores will be formed. Okay, through that pores we can introduce our gene of interest. Okay, you can see in this particular diagram, so this is the cell and these are the DNA fragments, and these DNA fragments and the cells are, they will be mixed together. Okay, and they will mix together and they are placed in a special cuvette. Okay, cell mixture as well as the DNA fragment. The DNA to be transferred are mixed together and kept in a special cuvette. So here what happens is, here you will having a cell as well as the DNA to be introduced, and then this cuvette is kept in an instrument where electric pulses will be given. Okay, the cuvette containing the cells and the DNA is placed in a electric instrument or an instrument where we can give the electric pulses. And then what happens is, it's like calorimeter only. So how you keep in your cuvette in a calorimeter, like that you have to keep the cuvette in a electrophoresion unit. Okay, so once the electric pulses are generated, see here the pores are being made. You can see the pore over here, and through this pore the DNA enter into the host cell. Okay, and the genes are introduced into the host cell. So this is the gene transfer by electrophoresion. Okay, second method of gene transfer is liposome mediated gene transfer. So liposomes are nothing but these are circular lipid molecules which have an aqueous interior region that can carry the nucleic acid. Okay, so second method of transfer is gene transfer is liposome mediated gene transfer. Okay, this will be done through the liposome. So liposomes are nothing but they are circular lipid molecules which have an aqueous interior that can carry the nucleic acid and several okay, and through this liposuction you can use it to transfer the genes into bacterial cells, animal cells and plant cells. Okay, the liposome mediated gene transfer is also referred to as liposuction. Okay, liposome mediated transfer is referred to as liposuction. Okay, so we will see how it will be done. Okay, so what happens is, on treatment of DNA fragment with liposomes. So liposomes are nothing but which will be having an aqueous interior. So on treatment of DNA fragment with liposome, the DNA pieces get encapsulated inside the liposomes. We have discussed that liposomes interior, they are made up of aqueous molecules. Outside it will be having the hydrophobic zone. Okay, so on treatment of DNA fragment with liposome, the DNA pieces get encapsulated inside the liposome because of the DNA is polar in nature. So because of the phosphate group, it can able to present in the aqueous solution. So DNA gets encapsulated inside